Welcome to the Creative Commons RX podcast. Before diving into this episode, I want to ensure we're all on the same page. This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this podcast is at the user's risk. The content on this podcast is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay in obtaining medical advice from any medical condition they have, and they should seek the assistance of their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the audio content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures.
Welcome to the Deep Dive. We're here to cut through the complexity of cutting-edge science and get straight to the core knowledge you need. Today, we're diving into something truly transformative: genetic medicine. It's really moved beyond just promise, hasn't it? It's actually here, in clinics, changing lives.
It absolutely has. It's a huge shift. We're not just managing symptoms anymore. We're talking about altering or adding or even correcting a patient's own genetic material. So our mission today is to really unpack the main strategies and the technologies behind these therapies, especially the ones given in vivo, directly inside the patient's body.
Okay, in vivo. Let's start there. If we're going inside the body to tweak the genome, what are the sort of main approaches? You mentioned altering, adding, correcting.
Right. Broadly speaking, there are two fundamental strategies right now. First, there's gene replacement, which is sometimes called gene addition.
Finger placement.
Okay. This is the principle behind, all the currently approved in vivo gene therapies using AAV vectors. The idea is pretty straightforward. If a person has a gene that's not working because of say, a loss of function mutation.
Right, the gene's broken.
Exactly. So we introduce a new functional copy of that gene. We don't actually touch the faulty one that's already there. We just give the cell an extra working blueprint.
So it's like slipping a correct instruction sheet into the binder, even if the old torn one is still in there. The cell can now read the new one to make the protein it needs.
Precisely. It provides a supplementary template. Now, the other strategy is gene editing.
And this sounds more surgical.
It is. It's aiming for much greater precision, the next frontier, really. The goal here is to make specific, permanent changes to the existing DNA sequence right at the source.
So not just adding a copy, but actually rewriting the original text?
Exactly. This could mean correction, like fixing a typo, repairing the mutation back to the normal sequence. Or it could mean knockout, where you intentionally disable a gene that's causing problems, maybe producing a toxic protein, or even knock-in, where you insert a new piece of DNA at a very specific spot in the genome, much more targeted.
Got it. Replacement adds a functional copy. Editing changes the original sequence. But for either of these to work in vivo, you need to get the goods, the gene or the editor, into the right cells. That brings up delivery, right?
Yeah.
In vivo versus ex vivo.
A really critical distinction. Our focus today is in vivo, which means we administer the therapy directly into the patient. Think 5E infusions, or maybe a very localized injection, like into the eye or muscle.
And the genetic modification happens right there, inside the body.
Correct. This is pretty much essential if you need to target internal organs that are hard to get at, otherwise the liver, the brain, the central nervous system, the eye. It's also, potentially more scalable for treating large patient populations.
Okay, but what about ex vivo? We hear about that too, especially for blood disorders.
Right, but ex vivo, you take cells out of the patient, often blood stem cells, hematopoietic stem cells. You then engineer them in the lab, making the genetic modification there.
Where you can check your work, presumably.
Exactly. That's the big advantage. You have much more control. You can verify the edit or the addition worked correctly and safely before you infuse those modified cells back into the patient.
But it sounds complicated.
It is. It's complex, it takes time, it's expensive, and generally less scalable than a direct in vivo injection. So, for our deep dive today on in vivo therapies, especially the gene replacement ones, we really need to talk about the delivery vehicle that makes it all possible. The workhorse.
The adeno-associated virus, AAV.
That's the one. AAV is really quite remarkable. It's this tiny, tiny virus, only about 25 nanometers across. It's a parvovirus, naturally occurring, but crucially, it doesn't cause disease in humans.
And for therapy, we're not using the wild virus, are we?
Oh, absolutely not. We use recombinant AAVs, or rAAVs. We basically gut the virus. We take out its own genes, the ones it needs to replicate, like rep and cap. So the vectors we use are replication deficient. They can't make more of themselves. Very important for safety.
So you take out the viral engine.
And in its place, we package our therapeutic payload, the gene we want to deliver, along with the necessary control elements, like a promoter. That's the expression cassette.
So it's like a delivery capsule. What parts of the original virus do you keep? What's essential?
The only viral bits we absolutely need are the inverted terminal repeats, the ITRs. These are short DNA sequences found at both ends of the AAV's natural genome.
And they're critical because
They do two key jobs. First, they act like packaging signals, telling the cellular machinery to stuff our therapeutic DNA into the AAV capsid shell. Second, and this is crucial for how the therapy works long-term, they play a vital role once the vector gets inside the target cell's nucleus.
Okay. So what happens in the nucleus? This is where the magic of long-term expression happens, right?
Precisely. The AAV delivers its payload, which is single-stranded DNA flanked by those ITRs. Once in the nucleus, the host cell's own machinery recognizes the ITRs. It synthesizes the complementary DNA strand.
Make it double-stranded.
Right. And then, often through mechanisms involving those ITRs again, this double-stranded DNA molecule circularizes. It forms a stable, independent circle of DNA called an episome.
An episome. So it's not integrated into the host chromosome.
Generally, no. That's a key feature. It sits separately in the nucleus. And this is why AAV works so well in certain tissues.
Which tissues are those? The ones that don't divide much?
Exactly. Think neurons in the brain, muscle cells, photoreceptor cells in the retina. These cells are postmitotic, or they divide very, very slowly. In these cells, that episome just persists.
It doesn't get diluted out every time the cell splits.
Correct. So it hangs around, acting as a stable template, allowing the cell to continuously transcribe the therapeutic gene and make the needed protein for months, even years, potentially decades, all from a single dose. That's the basis for durable expression.
That's incredible, but also highlights a limitation, right? If the target cells are dividing rapidly.
Then AAV-based replacement isn't ideal. The episome would get diluted and eventually lost as the cells proliferate. That's why it's not suitable for something like blood stem cells, which divide constantly, or for cancer cells. You need different approaches there, often ex vivo or different vectors.
Okay, so durable expression in non-dividing cells. Now, AAV isn't just one thing. You mentioned there are different types, serotypes.
Yes, a whole family. There are over 100 known AAV serotypes, plus many engineered variants now. Each serotype has a slightly different protein code, the capsid.
And that capsid determines where the AAV goes.
Precisely. The capsid structure dictates the virus's tropism, its natural affinity for binding to and entering specific types of cells or tissues. Choosing the right serotype is absolutely critical for getting the therapy to the right place.
So you tailor the delivery truck for the specific address. Can you give some examples from the therapies that are actually approved?
Sure. Look at Luxturna for inherited retinal dystrophy. It uses AAG2.
No.
Why? Because AAV2 is known to be very efficient at transducing or getting into the retinal pigment epithelial cells, the RPE cells, which is the target.
Makes sense. Target the eye, use an eye-loving AAV. What about getting into the brain across the blood-brain barrier? That's famously difficult.
Extremely difficult. That was a major challenge for Zolgensma, which treats spinal muscular atrophy, a neurological disorder affecting motor neurons in the spinal cord. They needed systemic delivery, an IV infusion that could reach the central nervous system.
So which serotype cracked that?
AAV9. It has this remarkable, somewhat unique ability among the common serotypes to cross the blood-brain barrier. It interacts with specific receptors on the cells lining the brain capillaries and gets transported across into the CNS. That was a game changer for treating neurological diseases in vivo.
Wow. And for something like Duchenne muscular dystrophy, affecting muscles all over the body.
For Elevides, which treats DMD, they selected AAVRH74. This serotype showed strong tropism for both skeletal muscle and cardiac muscle, which is vital because DMD affects the heart too. So the choice of serotype is a highly strategic decision based on the disease target.
Okay, so we have this durable system, customizable for different tissues. It sounds great, but we know there are big hurdles. What are the major limitations of AAV vectors?
There are two really critical ones we have to grapple with. The first is a fundamental physical constraint, the limited packaging capacity.
How limited are we talking?
Very limited. An AAV capsid can only hold about 4.7 kilobases, kb, of single-stranded DNA. That's it. The physical volume inside the capsid just isn't big enough for anything larger.
4.7 kilobounds, and that immediately causes problems for certain diseases, right? Like DMD again.
Exactly. The gene that's missing in DMD patients, the dystrophin gene, DMD, is enormous. It's about 11 kilobases long in its coding sequence alone.
More than double the AAV capacity, so you just couldn't deliver the whole thing.
Impossible with a single AAV vector. This limitation directly forced the field into a major bioengineering effort. It led to the creation of the microdystrophin used in Elevities.
Microdystrophin. How do you shrink down such a critical, huge protein and still have it work?
It was and is a remarkable feat of protein engineering. Dystrophin acts like a shock absorber in muscle cells, linking the internal structure, the cytoskeleton, to the outer membrane. Scientists had to figure out the absolute minimum essential parts.
What did they keep?
They kept the crucial ends, the N-terminus that binds to the cytoskeleton, and the C-terminus that links to the membrane protein complex. Then in the middle rod section, which is made of many repeating units, they kept only a few strategically chosen repeats and flexible hinge regions that are critical for its shock-absorbing function.
So they cut out a lot of the repetitive part.
A lot. They basically compressed the essential functional domains into a package small enough under 4.7 kilobatters to fit inside the AAV. It produces a shorter but still functional version of the protein.
A brilliant workaround driven entirely by the vector's limitation.
Okay, what's the second major constraint? This one's biological, right? In the patients themselves.
Yes, and it's a huge issue. Pre-existing immunogenicity. AAVs are common viruses in nature. Many people, a significant percentage of the population.
How many are we talking?
It varies by serotype, but estimates range from 40% to sometimes over 70% of people have already been exposed to some AAV serotype naturally.
And their immune systems remember it.
Exactly. They've developed neutralizing antibodies, NAbs, against the AAV capsid. If a patient has high levels of these NAbs,
The therapy gets intercepted.
pretty much immediately, the antibodies bind to the incoming vector, flagging it for destruction before it can even reach the target cells. The therapy would be ineffective.
So you have to check for this beforehand. Absolutely mandatory. NAbs screening is a required part of the clinical protocol for therapies like Elevities and the new hemophilia B therapy, BEQ-VEZ. Patients are tested for these antibodies, and if their levels are above a certain threshold, they're unfortunately ineligible for the treatment. It's a major barrier to access.
Wow. Okay, so you screen out patients with pre-existing antibodies, but even if a patient is NAb negative, introducing a large dose of virus particles, even if they're replication deficient, can still provoke the immune system, right?
Yes, particularly with systemic high-dose IV infusions. The body can mount an inflammatory response against the capsid proteins themselves. And this leads to the most significant safety concern observed with systemic AAV therapy.
Which is?
Hepatotoxicity, liver injury. The AAV vectors, especially after IV delivery, tend to accumulate significantly in the liver. This can trigger an immune attack on the transduced liver cells.
How severe can this get?
It can range from elevated liver enzymes, indicating stress, to acute serious liver injury, and in some rare but reported cases, even acute liver failure. It's a major risk that has to be managed proactively.
So how do clinical teams manage it? What are the standard protocols now?
It involves rigorous monitoring and immune suppression. Patients getting systemic AAV therapies like Zolgensma or Elevities require frequent blood tests to check their liver enzyme levels, aminotransferases, for weeks and months after infusion.
And immune suppression.
Yes, mandatory prophylactic use of systemic corticosteroids, like prednisolone. This usually starts before or on the day of the AAV infusion and continues for at least 30 days, sometimes much longer, depending on the therapy and the patient's response. The goal is to tamp down that immune response and protect the liver.
So it's clear these therapies aren't just about getting the gene in, they're also about carefully managing the body's reaction to the delivery vehicle. Let's maybe walk through the 4 big approved in vivo AAV therapies now. Each seems to represent overcoming a specific hurdle.
Sounds good. We should start with Luxturna, approved first back in 2017. It treats a rare form of inherited blindness caused by mutations in the RPE65 gene.
And this was the crucial proof of concept, right? That this could actually work in vivo in humans.
Absolutely foundational. The RPE65 protein is essential for the visual cycle in the retina. Without it, patients suffer progressive vision loss, often leading to blindness.
And the delivery strategy.
Highly localized. They used AAV2, known for hitting retinal cells, and delivered it via a one-time subretinal injection. It's a surgical procedure, injecting the vector directly under the retina.
Right into the target tissue. Advantageous because the eye is somewhat protected from the immune system.
Exactly. The eye is considered immune privileged. This means the immune response is naturally dampened there compared to the rest of the body, reducing the risk of inflammation rejecting the vector.
How do they actually measure if it worked? It can't be just reading an eye chart if someone is severely visually impaired.
Yeah.
They developed a really clever functional test called the Multi-Luminance Mobility Test, or MLMT. It's performance-based. Patients have to navigate a standardized obstacle course under very low light conditions,
Like how low?
down to 1 lux, which is incredibly dim, think of moonlight or less. In the key trial, 65% of patients treated with Luxturna could successfully navigate the course at the 1 lux level after a year, whereas none of the control group could.
Wow, a real functional improvement in navigating the world.
A durable one, too. It showed improvement in functional vision, not just a lab measurement. And the safety profile reflected the localized delivery. Most side effects were related to the surgery itself, like cataracts or retinal tears, rather than systemic immune issues.
Okay, so Luxturna proved it could work in an accessible, immune-privileged site. Then came the leap to systemic delivery for a devastating neurological disease. Zolgensma for spinal muscular atrophy, SMA.
Right. SMA is caused by mutations in the SMN1 gene, leading to a deficiency of the SMN protein, which motor neurons need to survive. SMA type 1 is the most severe form. Untreated infants typically die or need permanent ventilation by age 2, a truly horrific disease.
And the challenge was getting the therapy to those motor neurons throughout the spinal cord.
Hence the choice of AAV9, delivered as a single, one-time IV infusion, leveraging its ability to cross the blood-brain barrier. The goal was to deliver a functional copy of the SMN gene to those struggling motor neurons.
The results were pretty stunning, weren't they?
Paradigm shifting. We saw transformative improvements. Untreated SMA type 1 infants almost never achieve motor milestones like sitting up without support. In the Zolgensma trials, we saw dramatic improvements in survival without permanent ventilation, and many children achieved milestones previously thought impossible, sitting, crawling, some even walking.
That's incredible. And these effects lasted.
The data shows durability for years post-treatment. It fundamentally changed the natural history of the disease for treated children.
But this systemic delivery brought that major safety concern to the forefront, the liver toxicity.
Yes, the risk of acute serious liver injury became very clear. It underscores the trade-off with systemic AAV. That's why the rigorous liver enzyme monitoring and the mandatory prophylactic corticosteroids for at least 30 days became absolutely essential parts of the Zolgensma protocol.
Okay, next up, Elevidys for Duchenne muscular dystrophy. This one really highlights overcoming that AAV packaging limit we talked about.
Definitely. DMD is caused by the lack of functional dystrophin protein. The DMD gene is huge, 11 kilobands, far too big for AAV's 4.7 kilobeta limit.
So the solution was the bioengineered microdystrophin.
Delivered using the muscle-targeting AAVrh74 vector. Elevidys has had maybe the most complex regulatory journey so far.
How so?
Because functional improvement in DMD takes time to measure definitively, the FDA initially granted accelerated approval. This was based on data showing that the microdystrophin protein was actually being produced in the muscles of treated boys, a surrogate endpoint considered reasonably likely to predict clinical benefit.
But then the main clinical trial didn't quite hit its primary goal.
Correct. The Phase 3 trial, called EMBARK, which was designed to confirm the clinical benefit, did not meet its primary endpoint, which was the change in the North Star Ambulatory Assessment NSAIR score, a standard measure of motor function in DMD.
That sounds like bad news for full approval.
Normally it might be. But the FDA looked at the totality of the evidence. While the primary endpoint wasn't met, several key secondary endpoints did show statistically significant and clinically meaningful improvements. Things like how quickly boys could rise from the floor or their time on a 10-meter walk-run test.
So even though the main scale didn't show a difference, other functional measures did.
Yes, and based on that strong supporting data, the FDA converted Elevidys to traditional approval for ambulatory patients aged 4 and older, while also expanding the accelerated approval to include non-ambulatory patients. It was a nuanced decision based on the overall picture.
And the safety profile for Elevidys, besides the liver concerns.
Similar liver monitoring and mandatory steroid protocols are needed, often for about 3 months. But unique to Elevidys, there are also risks of immune-mediated myositis, muscle inflammation, and even myocarditis, heart muscle inflammation. So they also monitor cardiac troponin levels.
And there's a specific genetic exclusion.
A crucial one. Elevidys is contraindicated, meaning it must not be used in patients who have deletions affecting exon 8 and/or exon 9 in their DMD gene. Pre-treatment genetic testing for these specific deletions is required.
Why that specific exclusion?
Because clinical data showed these patients are at a significantly higher risk of a severe, potentially life-threatening immune reaction against the microdystrophin protein itself. It's a critical safety screen.
Got it. Okay, lastly, the most recent approval. BEQVEZ for hemophilia B. This seems like a story of optimizing the actual therapeutic gene being delivered.
That's a great way to put it. Hemophilia B is caused by a deficiency of factor IX, FIX, a crucial blood clotting protein. Patients typically need lifelong prophylactic infusions of FICUP concentrate to prevent bleeds.
And BEQV offers a one-time IV infusion alternative.
Yes, using an AAV vector targeted to the liver, where FIFX is normally produced. But the clever part is what gene they put inside the AAV. They didn't use the standard human F9 gene.
What did they use instead?
They used a naturally occurring high-activity variant of the gene called FX-PADWA. This variant has a specific single amino acid change, R338L, that makes the resulting FX protein hyperfunctional.
Hyperfunctional, how much more active?
Significantly more active. The fixed PADWA protein has somewhere between 5 to 8-fold higher specific activity in promoting blood coagulation compared to the normal wild-type FX protein.
Wow. What's the therapeutic advantage of that?
It means you can achieve clinically meaningful levels of clotting activity enough to protect against spontaneous bleeds with potentially much lower levels of protein expression in the liver. And crucially, this might allow for the use of a lower vector dose compared to using the standard gene.
And a lower dose could mean less risk of that liver toxicity and immune response we keep talking about.
That's the potential benefit, yes. In the clinical trials, BEQVEZ was shown to be superior to the standard prophylactic FX infusions. The median annualized bleeding rate, ABR, dropped to 0 for treated patients, effectively offering a functional cure and freeing them from the burden of regular infusions.
That's a really smart optimization. So we've seen this progression: localized delivery, tackling systemic delivery and the baby B, engineering around packaging limits, and now optimizing the payload itself. It shows incredible innovation within the gene replacement framework using AAV.
Absolutely.
But now the field is increasingly looking towards the next step: gene editing. If AAV gene replacement is working, why the push towards editing? What's the fundamental draw?
The ultimate draw is the potential for a true, permanent correction of the underlying genetic defect at its source, rather than just adding a supplemental copy. Gene editing aims to fix the original blueprint.
And how does it work? What's the basic mechanism?
At its core, gene editing relies on using programmable nucleases. These are essentially molecular scissors that can be guided to a very specific sequence in the genome.
And they cut the DNA there.
Exactly. They create a targeted double-strand break, DSB, in the DNA backbone at that precise location. This break is the key event. It acts as a signal that triggers the cell's own natural DNA repair machinery.
So we're leveraging the cell's repair kit.
We're hijacking it, essentially, to make the change we want.
Okay, programmable nucleases. This technology didn't just appear with CRISPR, right? There was an evolution. What came before?
Right. The concept has been around longer. The first-generation platforms were zinc finger nucleases, ZFNs.
Fingers. Like the protein structures?
Yes. ZFNs are engineered proteins where an array of zinc finger domains provides the sequence recognition. Each zinc finger typically recognizes a 3-base pair DNA triplet. You stitch several together to target a longer sequence and then fuse them to a nucleus domain, like Foci, that does the cutting.
Sounds complex to design.
Extremely.
Yeah.
Engineering a ZFN array with high specificity for a new target site was very challenging, time-consuming, and often context-dependent. It wasn't easily scalable.
So what improved on ZFNs?
The next major platform was transcription activator-like effector nucleases, TALENs. These also use protein domains for DNA recognition, but the tail domains recognize single nucleotides, not triplets.
More like building blocks. One protein module per DNA base.
Exactly. This made the design principle much more modular and predictable than ZFNs. It was a significant step forward in programmability.
But still protein-based recognition. Any downsides?
They were still quite large proteins which could complicate delivery into cells. And while more predictable than ZFNs, designing and validating them still required significant protein engineering expertise.
And then came the system that really blew the doors open, CRISPR-Cas9. Why did CRISPR become so dominant so quickly? What was the fundamental difference?
The game changer was the guidance mechanism. ZFNs and TALENs rely on protein-DNA interactions for specificity. You have to re-engineer the protein for every new target. CRISPR-Cas9 is RNA-guided.
RNA-guided. How does that work?
The system uses a short piece of RNA, called the guide RNA, gRNA, which contains a sequence that is complementary to the target DNA site you want to cut. This gRNA essentially acts like an address label. It binds to the target DNA sequence through simple Watson-Crick base pairing.
And it brings the scissor with it.
Exactly. The gRNA complexes with the Cas9 protein, which is the nuclease, the enzyme that actually makes the double-strand break. So the Cas9 protein is directed to the specific DNA site by the guide RNA.
So to change the target, you just change the RNA sequence.
Precisely. Instead of needing complex protein reengineering, you just need to synthesize a new guide RNA with a different targeting sequence. This is vastly simpler, faster, and cheaper. It democratized gene editing, making it accessible to labs worldwide. The specificity shifted from hard-to-engineer proteins to easy-to-design RNA.
That makes total sense. Huge difference in usability. Okay, so CRISPR-Cas9 makes the targeted cut, the DSB. Then, as you said, the cell's repair machinery kicks in. What happens next? How does that lead to an edit?
The outcome depends entirely on which of the cell's two major DNA repair pathways gets used to fix that break.
Two main pathways, what are they?
The first, and the one that's most active in mammalian cells, is called non-homologous end joining, or NHEJ.
Non-homologous. Sounds a bit messy.
It is. NHEJ is the cell's quick and dirty repair system. It basically just grabs the two broken ends of the DNA and sticks them back together as fast as possible.
Does it do it accurately?
Often not. NHEJ is inherently error-prone. As it joins the ends, it frequently introduces small, random insertions or deletions of DNA bases right at the cut site. These are called indels.
Insertions or deletions? That sounds like it would mess up the gene sequence.
It often does. If these indels occur within the coding sequence of a gene, they can cause a frameshift mutation. This scrambles the downstream genetic code, usually leading to a premature stop codon and the production of a truncated, non-functional protein.
Ah. So even though it's error-prone, you can use NHEJ intentionally.
Absolutely. If your therapeutic goal is gene knockout to disable a faulty or toxic gene, then inducing a DSB and letting NHEJ fix it with indels is exactly what you want. You're exploiting his sloppiness to break the gene.
Clever. Okay, so NHEJ is for knocking things out. What if you want to make a precise correction, fix a typo, not just break the gene?
For that, you need the cell to use the other major repair pathway, homology-directed repair, or HDR.
Homology-directed.
Sounds more precise.
Much more precise. HDR is a high-fidelity repair mechanism. The cell uses a template sequence, normally the sister chromatid, the identical copy of the chromosome present after DNA replication to accurately repair the break, ensuring the original sequence is restored perfectly.
So for therapy, we need to provide that template.
Exactly. If we want to achieve precise gene correction, like fixing a single base mutation or targeted gene knock-in, inserting a new sequence, we introduce the Cas9 and guide RNA along with an exogenous DNA donor template. This template contains the desired sequence flanked by regions that match the DNA around the cut site, homology arms, and the cell uses our donor template to fix the break accurately.
If HDR is active, yes, it uses the donor template as the blueprint to repair the DSB, incorporating the corrected sequence or the new insert into the genome at that specific spot.
Okay, NHEJ for knockout, HDR for precise correction or knock-in, but you said NHEJ is the dominant pathway. Does that create a challenge for achieving HDR?
A major challenge, especially in vivo. NHEJ is active throughout the cell cycle, while HDR is predominantly active only during the S and G2 phases when the cell is replicating its DNA and has that sister chromatid available.
And many of the cells we want to target, like neurons, muscle cells, aren't actively dividing, right? They're stuck in G0 or G1 phase.
Precisely. So in these critical non-dividing target cells, NHEJ is basically the only repair pathway operating efficiently.
Oh, wow.
This makes achieving high levels of precise gene correction via HDR in vivo in these tissues incredibly difficult. Getting knockouts via NHEJ is relatively efficient, but getting precise fixes via HDR is much, much harder.
So that's a big hurdle for corrective therapies in many diseases. Is research trying to, boost HDR?
Yes, it's a huge area of research. Strategies include temporarily inhibiting key NHEJ proteins to sort of force the cell towards HDR, or synchronizing cells, or developing newer editing tools that might be less reliant on these pathways, which we can touch on later.
Okay, that clarifies the molecular challenges. Now let's circle back to delivery. We established AAV is great for gene replacement because you want that durable episome. Why is AAV a bad, potentially dangerous choice for delivering gene editors like CRISPR-Cas9?
Because the durability of AAV expression becomes a major safety liability. if you used an AAV to deliver the gene encoding the Cas9 nuclease.
The cell would keep making the Cas9 protein indefinitely from that stable episome.
Exactly. You'd have a molecular scissor floating around in the cell, potentially for years. This persistent nuclease activity dramatically increases the risk of off-target cuts, the nuclease cutting at unintended sites elsewhere in the genome over time. The longer it's active, the higher the chance of collateral damage.
That sounds really risky. You could disrupt other important genes.
Or even activate oncogenes. So for gene editing, you absolutely need the editing machinery to be active only transiently. It needs to get in, make the cut, and then disappear. A hit-and-run approach.
So if AAV is out for editors, what's the preferred delivery vehicle for in vivo gene editing, especially for, say, liver-targeted therapies we're seeing in trials now?
The field has largely pivoted to non-viral vectors, primarily lipid nanoparticles, or LNPs.
LNPs, we heard a lot about those with the mRNA COVID vaccines. How do they work for gene editing?
They're ideal for transient delivery. LNPs are essentially tiny fat bubbles that can encapsulate nucleic acid payloads. For CRISPR editing, you typically package the guide RNA and, crucially, the messenger RNA, mRNA, that encodes the Cas9 nucleus inside the LNP.
So you're delivering the instructions to make Cas9, not the gene for it.
Correct. The LNP delivers the mRNA to the target cell, often the liver, as LNPs naturally traffic there. Inside the cell, the mRNA is translated by the ribosomes to produce the Cas9 protein.
And why is that transient?
Because mRNA is inherently unstable in the cell, it gets degraded naturally within a day or two. And the Cas9 protein itself also has a limited half-life and gets broken down by the cell's protein degradation machinery.
So the whole editing system is active only for a short period.
Exactly. The Cas protein is made, finds its target guided by the gRNA, makes the cut, initiates repair, and then both the mRNA instructions and the Cas protein itself are cleared away. This dramatically minimizes the window for potential off-target activity.
A much safer profile for something that permanently alters DNA. Are there other advantages to LNPs over AAV for editing?
Yes. Generally, LNPs are considered less immunogenic than AAV capsids, reducing concerns about pre-existing immunity or strong inflammatory responses. They also don't have the same strict packaging size limitations as AAV, which offers more flexibility for delivering larger or multiple components.
Okay, so that LNP-mRNA approach seems like the key enabling technology for moving in vivo editing forward safely. But even with transient delivery, making permanent cuts in the genome still carries risks, right? What are the main safety concerns specifically related to the gene editing process itself?
There are several critical ones. The first, as we touched on, is off-target effects. Even with a well-designed guide RNA, there's always a risk the nucleus might cut at other sites in the genome that have similar, though not identical, sequences.
And the consequences could be serious?
Potentially very serious. An off-target cut could disrupt an essential gene, or worse, happen near a proto-oncogene, and potentially contribute to cancer development down the line. Minimizing off-target effects is paramount.
How do researchers tackle that?
Through very careful guide RNA design, using computational tools to predict and avoid potential off-target sites. Also, by engineering high-fidelity Cas enzymes that are less tolerant of mismatches between the guide RNA and the DNA, making them more specific.
Okay, so off-target cuts, what else? Can things go wrong even if the cut happens at the right place?
Yes, those are termed on-target effects, but unintended ones. Even when the nucleus cuts precisely where intended, the subsequent repair process, especially the error-prone NHEJ, can sometimes lead to unexpected outcomes.
Like what?
We can see large deletions of DNA around the cut site, or complex chromosomal rearrangements, like inversions or translocations, especially if cuts happen on two different chromosomes simultaneously. These large-scale changes at the target site can also have deleterious consequences.
So ensuring the edit is clean, even at the right spot, is also crucial.
Absolutely. Requires careful validation and assays to detect these kinds of events.
And then there's the immune system again. We talked about antibodies against the AAV capsid. Can the immune system also react to the editor itself, the Cas9 protein?
Yes, and that's another major hurdle. Most commonly used Cas enzymes, like Cas9 from Streptococcus pyogenes, Strep pyogenes, are bacterial proteins. Humans can, and often do, have pre-existing immune responses or develop responses to these foreign proteins.
Both antibodies and T-cell responses.
Both. Antibodies could potentially neutralize the Cas9 protein before it even gets to work, reducing efficacy. T-cell responses could target cells that have taken up the editor and are expressing the Cas9 protein, leading to inflammation or clearance of the edited cells.
And this immunogenicity has big implications for using the therapy again.
Huge implications for redosing. If a patient mounts a strong immune response to Cas9 after the first treatment, it would likely make a second treatment with the same editor completely ineffective or even dangerous. It also complicates the idea of treating multiple different genetic conditions in the same patient over their lifetime using CRISPR-Cas9. Finding ways around this, perhaps using less immunogenic Cas variants or humanized versions, is critical.
This power to rewrite the genome permanently carries enormous ethical weight. We have to draw lines. What's the crucial distinction between the editing currently being explored in clinics and what's considered off-limits?
The fundamental line is between somatic gene editing and germline gene editing.
Okay, what's somatic?
Somatic editing targets the non-reproductive cells of the body, liver cells, muscle cells, blood cells, eye cells, etc. The goal is to treat a disease in that individual patient. Crucially, any genetic changes made in somatic cells are not heritable. They won't be passed on to the patient's children.
And this is what current clinical trials are doing.
Exclusively. Somatic gene editing for therapeutic purposes is widely considered ethically permissible, assuming rigorous safety and efficacy standards are met.
So what's germline editing then?
Germline editing involves making modifications to reproductive cells, sperm, eggs, or embryos. Any changes made at this stage would be heritable. They would be passed down through generations, becoming a permanent part of the human gene pool.
And the stance on that?
There is a very broad international scientific and ethical consensus against using germline gene editing for reproductive purposes at this time. The potential for unforeseen long-term consequences for future generations, the irreversibility, and the societal implications are just too profound. It remains firmly off-limits for making babies.
A critical boundary. Beyond the type of cell, there are huge societal ethics questions too, particularly around access, right? These therapies are incredibly complex and expensive.
Astronomical price tags, often millions of dollars per dose. This raises serious concerns about equitable access. Will these potentially curative therapies only be available to the wealthy, thereby widening existing health disparities? How do healthcare systems pay for this?
Are there solutions being discussed?
Yes, lots of discussion around innovative payment models. Things like outcomes-based agreements where a payment is tied to the therapy actually working, annuity models, spreading payments over years, risk pooling. It's a major challenge for society to figure out how to make these breakthroughs accessible. And related to that is ensuring truly robust informed consent, given the complexity and permanence of these treatments. Patients need to understand the risks and benefits deeply.
Absolutely. Now, looking forward, the technology itself isn't standing still. You mentioned tools that might get around some of the issues with the standard CRISPR-Cas9 cut.
Right, the field is rapidly advancing beyond just making that double-strand break. We're seeing the rise of next-generation editing tools, primarily base editors and prime editors.
Base and prime editing, how are they different? Do they still use Cas9?
They're derived from CRISPR-Cas9, often using a modified Cas protein, sometimes one that only nicks one DNA strand or doesn't cut at all, to guide them to the right location. But the key difference is they don't rely on creating that double-strand break to make the edit.
No DSB. How do they change the DNA sequence then?
Base editors work like a pencil with an eraser. They chemically convert one DNA base directly into another without cutting the DNA backbone. For example, one type can convert a CG base pair into a TA base pair. Another can convert an AT into a GC. They're great for correcting specific single-letter typo mutations.
Wow, direct chemical conversion. And prime editors.
Prime editing is more like a search and replace function. It uses a modified Cas protein fused to a different enzyme, a reverse transcriptase. It's guided by a special prime editing guide, RNA, page RNA, that not only targets the location but also carries the template for the desired new sequence. It nicks one strand, and the reverse transcriptase directly synthesizes the corrected sequence using the PEG RNA template. It can make small insertions, deletions, or correct multiple bases nearby.
So both base and prime editing avoid the DSB. What's the big potential advantage of that?
The major hope is enhanced safety. By avoiding the double-strand break, they largely bypass the cell's reliance on that error-prone NHEJ pathway. This should significantly reduce the risk of those unwanted indels, large deletions, and chromosomal rearrangements that can happen with standard CRISPR-Cas9 cutting. It potentially offers much higher precision and a cleaner edit.
Fascinating. More precision, maybe fewer side effects. That seems like a really important direction for the field. What an incredible journey we've traced today. From the early days of AAV, figuring out how to deliver genes in vivo with Luxturna.
Then tackling systemic delivery across the blood-brain barrier with AAV9 for Zolgensma.
Engineering clever workarounds like microdystrophin for Elevities to beat the AAV size limit.
And even optimizing the therapeutic gene itself with the fixed Padua variant in BQVAs for better potency.
It really shows this iterative process of identifying a problem, whether it's delivery, packaging, or payload, and engineering a solution within the gene replacement framework.
And that whole evolution really sets the stage for the current shift towards gene editing, driven by the precision of CRISPR and enabled by delivery systems like LNPs that offer transient expression.
The core insight, it seems, is that the entire strategy, the vector choice, AAV versus LNP, the expression pattern, durable versus transient, it's all meticulously dictated by the specific therapeutic goal and, crucially, by the safety considerations of messing with the genome.
Absolutely. Use durable expression, like AAV provides only when the long-term presence of the transgene is the goal and the risks are manageable. You absolutely must use transient expression, like from LNP-delivered mRNA, when the tool itself, the nucleus, would be dangerous if it stuck around.
The engineering choices reflect a deep understanding of the biology and the potential risks. It's an incredible balancing act. And that leads us perfectly into our final provocative thought for you, our listeners. We've spent a lot of time discussing how scientists have to manage the body's immune response to the delivery vehicle, the AAV capsid, using NAB screening and immune-suppressing steroid.
Constant battle against the host's defenses.
Right. But now consider this. We know the gene editing tool itself. The Cas9 protein is often bacterial and therefore immunogenic. Patients can develop immune responses to it. So what are the really profound long-term implications of that? If a patient needs their gene therapy topped up or re-administered years later, or if they develop a completely different genetic condition that also needs editing, how does the field overcome the challenge of the patient's immune system potentially blocking the very tool needed for the cure, especially if they've seen it before?
That challenge of redosing or treating sequentially due to Cas9 immunogenicity is a huge question hanging over the long-term future of in vivo editing. We might need entirely new, non-immunogenic editors or clever ways to cloak the current ones.
Something significant to ponder. Until next time on The Deep Dive, keep digging deeper.
This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this podcast is at the user's risk. The content on this podcast is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay in obtaining medical advice from any medical condition they have, and they should seek the assistance of their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the audio content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures.